Okay, so this is the first part of Lecture 3, which is about inheritance. And so for the first part, we're going to talk mostly about autosomal dominant and recessive inheritance. We will talk a little bit about X-linked inheritance, but more of X-linked inheritance will be in part 2 of Lecture 3. Okay, the first thing we want to do is we want to first talk about pedigrees. Now, pedigrees are simply charts or a diagram which are going to show the occurrence and appearance of phenotypes of a particular gene, and it's going to show them in relationship to ancestry. So your ancestors and whether or not they had those genes and how the genes got passed down through the family. And so first we have to talk about interpreting a pedigree chart. Many of you have seen pedigree charts. And so there's just a few rules, and we're just going to go very quickly through these rules. So the first two steps whenever looking at a pedigree is to determine two main sort of overarching questions. So first of all, you want to know, does the pedigree chart show autosomal or X-linked diseases? It can also show Y-linked disease, but most often it's either going to be autosomal or X-linked. So that's the question you want to ask when looking at the pedigree. Now, autosomal, of course, is going to be non-sex chromosomes, and your X and Y are going to be your sex chromosomes. So you're asking, is it in relationship to the sex chromosomes or not? So to answer this question is if most of the males in the pedigree are affected, then the disorder is most likely X-linked. If it is more of a 50-50 ratio or close to that, then both equal men and women having the disorder, it is most likely going to be autosomal. The second question is you want to determine whether it is going to be dominant or recessive. Dominant means that you only need one copy of the mutated gene in order to present with the phenotype. Recessive means you're going to need both copies of the mutated gene to present with the phenotype. So if the disorder is dominant, one of the parents must have the disorder. If the disorder is recessive, neither parent has to have the disorder, since they may be heterozygotes. Now, if we look at autosomal recessive, here's a pedigree showing autosomal recessive. You can see that you have both sexes have equal frequency. So the circles are the females, the squares are the males. And if you go through this, you can see that in general, there's equal representation of the phenotype within both male and females of this pedigree. In addition, the trait tends to skip generations. So if you look at this group over here, you can see that you do have somebody who is affected. That's that person, number one. However, each of their offspring, shown here, these three yellow, are unaffected. So it's skipping that generation, and then you see it pop up again in a next generation. So that is common with autosomal recessive as well. In addition, affected offspring are usually born to unaffected parents. So if you look at these two parents here, they are both unaffected, and they do have an affected offspring. So when both parents of the autosomal recessive are heterozygotes, then the approximately 1/4 of all progeny will be affected, and it appears more frequently among children of consanguine marriages. Consanguine means closely related marriages. We'll talk more about that a little bit later in the lecture. So now an autosomal dominant, again, has similar rules as recessive in that it has to appear with both sexes with equal frequency. So when you look at this, you can see that there's 4 male, 2 female, and that's close enough. It's a little bit off, but still it is showing up in both males and females. Both sexes can transmit the trait to their offspring. So you have a male who is transmitting, and here it's both the male and female who are affected, so they're both obviously transmitting. It does not skip generations, and so you can see that the affected generation passes it down to the next. The unaffected generations are, of course, not going to transmit the trait because there is no hidden. It's not recessive. It's not hiding in the genotype. If it is present, it's going to present in the phenotype. So this is where if you are an unaffected, that means you do not carry the actual trait. Affected offspring must have an affected parent unless it's a new mutation. So, and a new mutation wouldn't look like this. You wouldn't have generations of presentation of the disease. You would just have a single presentation of the disease in the family history. And then, of course, when one parent is affected, the heterozygote is more common than a homozygote. The other is unaffected. You get about half the offsprings will be affected. And that's showing kind of in this group over here. Now with X-linked is going to have, if it's dominant, it's going to have, again, both males and females affected. And in this case, often you're going to find more females than males. It also does not skip generations. All affected sons must have an affected mother. Affected daughters must have either an affected mother or an affected father. All affected fathers will pass the trait to all of their daughters. So here's an affected father, and all of the daughters get the trait because they can only get one X from the father. That's all he has. And it is an affected X, and so it will get passed down to every single daughter. Affected mothers, if heterozygote, will pass the trait on to one half of their sons. So here is an affected mother, and half of the sons of that mother are affected, for example. X-linked recessive is going to have more males than females will be affected, and that's because females have two X's, males have one, so if they get the affected X, that's the only X they have, and so they will present with the disease. Affected sons are usually born to unaffected mothers, so that would be a heterozygous, and so you could tend to have skipping of generations with this particular trait. So for example, you have parents 1, 2 up here, and they have an affected son here, so that would indicate that the mother is a carrier. Now, all the other children of the second generation do not have the trait. However, if you look at subsequent generations, the trait pops up again. So that means that this recessive trait is passing down from generation to generation, and it only presents itself when there is no alternative. For example, I'll add a carrier. So this one is a carrier. We have a carrier here, passed on, became a carrier here, and this son became affected. So here's an affected son. And the same here, we have a carrier, and so the son became affected here. Now you'd have to wait to see if this number 6, for example, is also a carrier. If she has a son, for example, or multiple sons, it could present. And again, all daughters of affected fathers are at least carriers. Okay, so rules of inheritance with Y-linked and mitochondria. Y-linked is pretty straightforward. Only males that have a Y chromosome are going to be affected, of course, so there are no females that are going to be presenting with the phenotype. It's going to be passed from father to sons, all sons, and it's because there's only one Y. It does not skip generations either. And then mitochondria, the trait is inherited from the mother only. All children of a mother are at risk to be affected or carriers. One of the things about mitochondria, it also looks like an autosomal, almost autosomal dominant, and the way you can see that it's not is because fathers do not transmit the disease. In an autosomal, you would get transmission from both mothers and fathers. In a mitochondria, transmission is only going to be my mother's. So if you look at this affected here in second generation number 1, this is a male who does not transmit the disease to any of their offspring. Here's an affected, here it's a female, and they transmit to their offspring. And you can go across and see all the males do not transmit to their offsprings, but the affected females will transmit. Okay, so now we're going to move into chapter 4. So we're going to talk about different diseases. We're going to define consanguinity, and then we're going to move into part 2, which will talk about sex-linked or X-linked diseases. And so we've already seen this Punnett square type approach, and Mendel generated this to show dominance, recessive, etc. And so what we're really interested in genetics, medical genetics, is how does the genotype and phenotype, how are they related? So I'm just going to talk a little bit about two diseases. The first is cystic fibrosis, and the second is phenylketonuria. And so in general, you have a genotype, and for cystic fibrosis, you're going to have mutations. This is going to be a autosomal recessive. So you have to have mutations on both alleles. So this is autorecessive. And then you also, for PKU, you also have to have the presence of mutations on both alleles. So it is also autosomal recessive. So what's different is going to be the phenotype. Okay, so the presentation of the phenotype is going to have three different components. And it's going to be influenced by the clinical component, the severity of the disease, and it also has a time course. Most affected with cystic fibrosis are going to be at a very young age. This also has linked to it consanguinity. And this is basically close relatives. So descendants of close relatives or kinship. And this is European. And then phenylketonuria is going to be where you have the same mutation does not equal the same disease. With phenylketonuria, you have something called modifiers. And these are genes that are going to express affect the expression of other genes. And so consanguinity has been shown to link to these modifiers, mutation in modifier genes that can result in different phenotypes within the phenylketonuria disease. And this has kinship also, and it's mostly in the Middle East. So there are two different diseases that evolved in two different areas of the world, and they both have similar aspects to them, especially with respect to consanguinity, because they're both going to be autosomal recessive. And so autosomal recessive traits are very rare unless there is some form of consanguinity. Okay, so histology 101, what structure is this? And here we're looking at, hopefully everybody can see these lovely isolates, the pancreas. So here's a healthy pancreas. And this is a cystic fibrosis pancreas. So cystic fibrosis, the chloride channels are important not only in the lungs, but they're also important in the pancreas and the gut. So individuals with cystic fibrosis are going to present with symptoms, respiratory as well as hormonal, your insulin and glucagon and things like that, as well as digestion. And so what is this structure? And here we have a healthy lung. And here we have a cystic fibrotic lung. And you can see that it has a lot of fibrosis associated with it. So cystic fibrosis is going to be caused by many different reasons. There are a lot of genes that have been associated with this disease. And so you can see that they have different classes. We have class 1, class 2, class 3, class 4. Here's class 5, class 6. Okay, that's good. So let's just take each of these apart. So let's start with class 1. So for class 1, there is no gene expression. And the gene we're talking about is the protein that codes for the receptor. So this is the chloride receptor on the surface of the cell. And so if you don't have expression of this receptor, obviously you're going to present with the disease. So the second type of mutation that can occur is that you do get plenty of your message made, but then what happens is your protein gets targeted for degradation. So in class 2, shown here, you make your message, you make your protein, but there's increased protein targeted to the proteasome. And then you get the protein breaking down. So what this means is that you have a functional actual receptor that's made. Some of it will get to the surface, but a lot of it will get degraded in the proteasome. So it's not going to be as severe as class 1, but you still don't have enough of the receptor on the surface for normal lung functions. Now number 3 is up here, and number 3 is associated with an abnormal regulation of chloride. So it doesn't turn on fast enough, or it doesn't turn off fast enough, so you don't get the right chloride regulation across the cell membrane. And then number 4, which is here, is abnormal chloride conductance. And so again, this has to do with the chloride moving across or not across. And so this is just going to affect how much of the function of the regulator, the chloride regulator, is present. Number 5 is back here in the nucleus, down here. So number 5 is where you have promoter mutations, or you can have intron-exon splice mutations. But basically, these are affecting protein synthesis because you have a decrease of messenger RNA, at least messenger RNA that codes for the actual true protein. You're going to have defective messenger RNAs depending on where this mutation is. And then in 6, shown up here, you have what's called accelerated turnover. So you have an increase in CFTR turnover. And so what turnover is internalization and degraded. So what that means is the cell has to continuously make more to keep up with the actual function of that receptor. And so you can imagine here that you're going to have different presentations of the disease depending on which class of mutation of the gene is going to affect which of these components. And so there are a lot of genes involved in making the message, making sure the message gets packaged and appropriately integrated into the membrane versus going to the proteasome. Accelerated turnover, so bringing the receptor back in, etc. So there's a wide variety of diseases associated with cystic fibrosis because of these different classes. So cystic fibrosis is going to overall increase the amount of chloride in the sweat. So you can do a chloride sweat test. Okay, and then you have a 95% sterility of males. So they cannot have offspring. There's over 2,000 known mutations, which leads to the variety of presentations of this disease. There are some treatments, and so the treatments are drugs that are going to allow your ribosomes to read through stop codons. And this is because there is a very common mutation, it's a missense mutation at position 551 that results in a stop codon. And so by having this drug, a lot of people, or majority of people, have this mutation, and it allows them to make a functional protein. And squalene at position 551. Okay, so that's cystic fibrosis. And so now let's talk a little bit about some other diseases. So we have some pedigrees. We've talked a little bit about pedigrees. And you can see here you have polydactyl, which is an autosomal dominant. And hopefully you can see that. You can see that you have both males and females are going to be affected. It does not skip a generation. The affected offspring have an affected parent. An unaffected parent will not transmit the disease to their offspring. So it's not a trait that can hide from generation to generation. So this autosomal dominant inheritance pattern is going to be very rare in populations. You're always going to have an affected heterozygote, usually they're not a homozygote. And then the probability is 50% of passing this trait on to their offspring. This results in a lack of skip generation. And then, of course, as you said, we have father to son transmission, and males and females are going to be affected. Autosomal recessive inheritance pattern, on the other hand, are going to be rare in populations as well. The heterozygotes are unaffected. They are also going to be more common than homozygotes, so you can have a trait hidden in a family pedigree. If you have an offspring with the phenotype, usually both parents are carriers. Most often, the parents are not affected, and oftentimes they don't even know they have the gene until they have an offspring who presents with the actual trait. You have clustering of these phenotypes among siblings, and consanguinity may be present if this autosomal recessive is prevalent in a family tree. So here is an autosomal recessive inheritance pattern of albinism. And so you can see here that we have a male up here who is a carrier. And the male passed on its gene to a female and another male. And the female passed on their gene to their offspring. The male passed on their gene to their offspring. This is consanguinity. It means close relatives had offspring. And because this trait was passing down through the family tree, even though they had no affected offspring up until this generation down here, because of consanguinity and close kinship interrelationships, they had two affected offspring. Okay, so recurrence risk is a risk for producing a child and the risk that future children will be affected. For autosomal dominant, you have typically one parent is going to be affected and one parent is unaffected. And so because that one parent is affected, they will pass it down to half their offspring. The other unaffected parent doesn't influence this risk because they don't carry the disease, and so it stays as one half. One thing to keep in mind is each birth is independent event. So you could, for example, if they only had 2 children, both could be affected. If they had more children, it would balance more out to 50-50. Autosomal recessive are most going to be 2 heterozygote carriers. And so that's going to lead to a risk of about a quarter for a homozygote. You could have a quasi-dominant state in which you have homozygotes that are going to mate with a heterozygote, and then that's going to bring up the risk to about 1/2, which is going to be similar to autosomal dominant. So just as a comparison, we're going to talk about usual recurrent risk for autosomal dominant is typically 50%. Very rarely do you have two affected that are going to have children. Autosomal recessive is typically going to be 25%. Again, it's usually because you're going to have both parents as heterozygotes. Usually a parent who is presenting with the disease is not going to be hopefully married to another person presenting with the disease in autosomal recessive, for example. The transmission pattern is going to be vertical. The disease phenotype is seen in generation after generation with autosomal dominant. Whereas in autosomal recessive, you're going to have your phenotype can be seen in multiple siblings, but usually not in earlier generations. So it's going to just pop up, but it is recessive. And so in that affected generation, it can be compounded just simply because of the higher prevalence between the mother and father of having those recessive traits. The sex ratio is equal numbers of affected males and females. It doesn't change between because we're autosomal, so dominant or recessive, you should have equal numbers. And then other notes are father to son transmission is possible. So this will be one way to determine if it's X-linked or autosomal, if you see father to son transmission. And for recessive, a lot of times it's consanguinity. All right, so there are going to be some exceptions. So most dominant diseases are going to be more severe than recessive in the heterozygote and non-viable in the homozygote. So if it's an autosomal dominant, more than likely an individual is a heterozygote because a homozygote would not survive. Achondroplasia is a disorder of bone growth. The heterozygotes are going to have short stature and live about 10 years less than normal. The homozygote is going to die in infancy from respiratory failure. Now, there are also some diseases that are inherited in either a dominant or recessive fashion, and it just depends on the mutation. So familial isolated growth hormone deficiency, IGHD, causes reduced stature. The gene is on chromosome 17. So you can have a recessive transmission pattern, and this is going to be a mutation, frameshift, nonsense, splice site mutation, which is going to result in a loss of function of that gene. So the heterozygote produce enough to be normal stature, and a homozygote don't produce any and have reduced stature. You can have a dominant transmission pattern, and this is by a dominant negative mutation of that growth hormone gene. It's a splice site mutation which deletes a third exon, and this leads to an abnormal protein, and this abnormal protein has the additive effect of disabling the normal protein. So that's why it's inherited as a dominant, autosomal dominant pattern, because even though you do have a good gene, the abnormal protein will inactivate the normal one. We have also beta thalassemia. This could be inherited either as dominant or recessive, and most are going to be autosomal recessive. The dominant is going to be due to a nonsense or frameshift mutation, and that's going to terminate translation in exon 3. So you get an unstable beta globulin chain, which has a dominant negative effect on normal chains. If the mutation is going to occur in exons 1 or 2, the result is very little abnormal messenger RNA is made, and the heterozygote is unaffected. But if it goes out to exon 3, you're going to have a lot of the mutated beta chain made, and it's going to incorporate into the normal hemoglobin, and it's going to have a negative effect on the function of the molecule. All right, so now there are different things that can express or can affect the expression of diseases. So here, for example, we have an embryo, and one of the cells in the embryo has a mutation. And so the offspring of this mutated cell will continue to divide in the embryo and will generate daughter cells that contain the mutation. And then as these daughter cells start to differentiate into different organs and organ systems, the organism has a mosaic of the mutated and non-mutated cells. So again, depending on the mutation, depending on the location of where these mutated daughter cells reside, what organ they eventually become part of, will affect the presentation of the phenotype. So we can have new mutations. That's pretty straightforward. New mutation just means it arises spontaneously. We have germline mosaicism. And germline mosaicism is where you have all a part of a parent's germline is affected by the disease mutation, but not all of the somatic cells. So that's sort of showing over here the embryo. The recurrence risk for offspring of mosaic parents is high. Type 2 osteogenesis imperfecta, achondroplasia, neurofibromatosis type 1, Duchenne muscular dystrophy, and hemophilia A are all examples of a germline mosaicism. Another property that can affect expression is something called reduced penetrance. And so reduced penetrance means that you have a trait that shows an autosomal dominant. So here you have an affected child, they have affected children, and so it's going through all the generations. So this is classic of an autosomal dominant. However, in this individual, they did not present with the actual disease, but they do have affected offspring. And so this is reduced penetrance. An example of reduced penetrance is retroblastoma. In this case, the person has the disease-causing genotype, but does not have a phenotype. And that's this AA in generation 2, has the disease-causing genotype, but does not have the phenotype. But they're going to pass the trait on to the next generation. So their obligate carriers are about 10%. So this AA is that 10% obligate carriers. They have the genotype, so they're carrying it with them. And penetrance is 90%. So most of the time you get penetrance of that genotype, but 10% of the time they become what's called obligate carriers, and it skips a generation. So retinoblastoma, again, is going to be reduced penetrance. This is going to be caused by mutations in the RB alleles, the retinoblastoma gene. You're going to typically inherit one allele, and then as you go through life, and you're going to have somatic mutations to the second allele, which occurs 90% of the time in people who have inherited one mutated allele. Retinoblastoma can also arise by a new mutation, so it can be not in the family pedigree, but it just occurs as a spontaneous new mutation. Retinoblastoma is going to bind to E2F, which is going to help prevent cell cycle progression. So retinoblastoma is a tumor suppression gene. And so what that means is if you have a loss of function, this leads to cancer. Because a loss of function means that the retinoblastoma can no longer bind to E2F, and then so E2F is free to have cell cycle progression and replicate. Penetrance is going to be all or nothing. So it's either you have the mutation and you are going to have the disease, or you're a carrier and you do not have the disease. So RB control of E2F, just very quickly, you have retinoblastoma. This is just a protein. You can think of it as a little helmet. And inside of the helmet is going to be E2F. So E2F is inside of the helmet. And so retinoblastoma is causing E2F to be non-functional. And then you're going to have cyclin-dependent kinases, and these are going to phosphorylate retinoblastoma. Sorry, some black. And you have phosphates, and then you get RB phosphorylated. And this releases the E2F protein. And now E2F is active. E2F is a transcription factor. And so this will go into the nucleus. Oops, and E2F will turn on genes. And the genes, of course, are going to promote growth, cell cycle, etc. And so retroblastoma prevents that. If retroblastoma is lost, and again, people who inherit the mutation already have one defective retroblastoma, 90% of the time, the second retroblastoma is going to become mutated, so they lose control of that gene. Now, other things that can affect expression of diseases with genes are something called age-dependent penetrance. All right, so age-dependent penetrance is basically what you're looking at is the frequency of the disease increases as the individual's age. So you can see that very low frequency of disease at an early onset, and as you age, the frequency increases. One of the diseases associated with this is hair loss. And so the follicles are normally going to go through different stages of growth. Then they do involution, quiescence, regeneration. Anagen, or the growth phase, is going to last for 3 to 5 years for a hair follicle. And then it's going to move into the catagen, which is the end of its growth. And then it's going to move into telogen, where it becomes quiescent. And then after quiescence, you're going to have regeneration of new growth. Androgenic alopecia is going to be hair loss, and it's most common male hair loss, affecting 30 to 50% of men by age 50. So what's happening with this is that you get hair follicle miniaturization, which is a hallmark of this, and it occurs in a progressive fashion, where there's greater than 10-fold reduction in the overall cell numbers producing hair follicles. The decrease of these numbers is not really known. It is associated with the androgen receptor gene on the X chromosome. However, you see this pattern from father to offspring, sons, and so it can't only be X-linked. There are other genes on your other chromosomes that influence the androgen receptor gene, and so those other genes play a role in whether or not people will present with this condition. Now, another is not only age affecting the disease expression, but there's also another term called penetrance and anticipation. So what basically this is you have late onset, which is going to be your penetrance of the disease. However, if it also has anticipation, it can shift to early onset. So anticipation is, for example, in Huntington's disease. So Huntington's disease was mapped to chromosomes using a process that identified the mutations in that chromosome, and they found at that site GAG repeats. Now, in a normal individual that does not have Huntington's disease, this CAG repeat is about 10 to 26 times in a row. If you have Huntington's disease, you are going to have different levels of amplification of this repeat. You can have anywhere from 27 to 35, and this is going to be larger than normal, but you don't have any disease. However, it does get passed on to your offspring, and this is where anticipation comes into play, because now your offspring are starting off already with a larger number of repeats. And so if they add to that, they can present with disease at a much earlier age. If the repeat is at 36 to 40, This is called incomplete penetrance. So you can have the disease. A lot of people will not have the disease when you're at this number of repeats. Greater than 40 is where you start to see the disease present all the time. And then the age of onset is going to be related to the number of repeats. 80% of all juvenile Huntington's disease, which is defined as presenting with the phenotype before 20 years of age, is associated with large repeats and paternal transmission. All right, so another term that's used, or that it plays a role in expression, is variable expression. This is going to impact the severity of the disease. It's not an all or nothing thing like penetrance. So we have autosomal dominant neurofibromatosis type 1, and then you have the environment, which can impact the expression of the disease, such as your exposure to tobacco smoke, diet, exercise. Also, modifier genes could have different impacts on the presentation of the disease. The different types of mutations for the same disease, so there's allelic heterogeneity. So depending on where the actual mutation is, can lead to differences in the gene. Allelic heterogeneity is also seen in the beta globulin mutations for sickle cell disease and your thalassemias. So here's variable expression of the neurofibromatosis. You have an autosomal dominant. So this is going to lead to different types of phenotypes. So less severe symptoms are going to lead to café au lait spots on your surface, shown here. You can have hyperpigmented patches. You can have nodules form, neurofibromatosis, which are going to be nerve cell tumors. Or you can present with more severe symptoms. So you can have a lot of neurofibromas, optic gliomas, learning disorders, different types of malignancies. Penetrance for neurofibromatosis is 100%, but the disease is going to have variability in the actual phenotype, the expression of the phenotype. Cystic fibrosis that we already talked about, there's different types of mutations that can lead to different phenotypes of cystic fibrosis, so the expression is going to be variable. You have partially active chloride channels or complete absence of chloride channels, so that's that type 1, complete absence. You also have other genes that can get involved. You have modifier genes that are going to increase expression, transcribing growth factor beta 1. You also have another modifier gene, dynactin protein function, and that's going to be involved in clearing bacteria from airways that can help exacerbate the disease if that one is also mutated. So again, depending on what genes, if it's just cystic fibrosis receptor, or if it's going to be also compounded by these other mutations in other genes. So there's also our myotonic muscular dystrophy. This is a phenotype that presents with myotona, which is an inability to quickly relax a muscle. You also can develop cataracts, narcolepsy, frontal balding, infertility, etc. Conditions expressed in individuals can be within the same family can be different. So here I'm just going to quickly draw a sort of a family tree with myotonia kind of going through the family tree. So you have here a father and then a mother, and the father is going to be affected, and the father has cataracts. Okay, and then they're going to have children, and so they'll have an unaffected daughter, they'll have an affected son, and this affected son is going to have myotonia, cataracts, and frontal balding. Okay, and then they're also going to have another unaffected son and a daughter, And this daughter will have myotonia and arrhythmia. All right, so this son does get married, and they have children. They have an affected son, and this affected son is going to have frontal balding and arrhythmias. They have an affected daughter who has cataracts. They have an unaffected son, and they have another affected daughter who has myotonia, and cataracts. So this is just to show that you have multiple people affected by this disease. However, their phenotypes are very different. So the father, the original, had only cataracts, and the offspring of the father had a variety of conditions associated with this actual disorder. Okay, so there are a few other things that can express. So one thing is locus heterogeneity. Locus heterogeneity is when you have mutations at multiple genomic loci, so different genes, are capable of producing the same phenotype, and each mutation is sufficient to cause the same phenotype independently. Okay? So that means you have gene X and gene Y. They're completely different. They're on different locations. Mutation of gene X leads to phenotype A. Mutation of gene Y leads to phenotype A. So regardless that they're different genes, different mutations, the outcome or the presentation of the disease is the same. Allelic heterogeneity is a single phenotype can be produced by multiple mutations all at the same locus of a chromosome. So that means you have a single gene, cystic fibrosis receptor gene, for example, that can be mutated at many points and lead to the same phenotype. Phenotype heterogeneity is where you have different phenotypes arise with identical genetics and environment conditions. And that was what we were discussing earlier, where you can have the same mutation but different presentation of the disease, and that different presentation from PKU, for example, phenylketonuria, is going to be caused by different effects of different modifier genes, for example. So you have the same mutation and you can have different phenotypes. So locus heterogeneity, examples of this are retinitis pigmentosa, hypertrophic cardiomyopathy, osteogenesis imperfecta, familial hypercholesterolemia, breast colon cancer, and melanomas. There is adult polycystic kidney disease, APKD, which is an autosomal dominant disorder. This one you have progressive accumulation of renal cysts, and you can have other cysts as well as hypertension and cardiac valvular functions, etc. Mutations in genes on either chromosome 16, PKD1, or chromosome 4, PKD2, will lead to the same phenotype. These encode membrane-spanning glycoproteins that are going to interact with each other, so losing one, you have the same phenotype as if you lost the other. And so these are going to regulate cell growth and signaling, so they are tumor suppressor genes, and so mutations in them will lead to overgrowth and the formation of cancers. Osteogenesis imperfecta is brittle bone disease. This is going to affect mostly the bone. The severity can be mild to severe. You can also have blue tinge to the whites of the eye, short height, hearing problems, breathing problems, problems with teeth. And these are always going to be associated with connective tissue. We talked about this already and talked about the different types. Most of it is due to lack of collagen type 1. These are autosomal dominance or mutation or a new mutation. And we talked about these. They could have collagen 1A1 mutations, collagen 1A2 mutations, and they will lead to a similar phenotype. Retinitis pigmentosa or rod cone dystrophy is going to have damage to the light-sensitive cells of the retina. This leads to severe vision impairment, decreased vision at night, so you get like a tunnel vision. There's no cure. There have been identified many mutations over 60, and the inheritance pattern can be autosomal dominant or autosomal recessive or X-linked. The genes that are involved are rhodopsin, retinitis pigmentosa, GTPase regulator, and also the gene for retinitis pigmentosa 2 protein. So mutations in all of those can lead to retinitis pigmentosa. Pleiotropy is where you have genes that have one or more effect on the body, so they have pleiotropic effects. The phenotype is pleiotropic. Marfan syndrome is autosomal dominant. There's an unusual stretching of connective tissue. The mutations are the genes that encode fibrillin. Fibrillin, of course, is found in many areas, so you're going to have effects on the eye, cardiovascular system, your skeletal system. Cystic fibrosis is also pleiotropic because you have effects on lungs, pancreas function, as well as sweat glands. Osteogenesis imperfecta, again, pleiotropic. You have bones, teeth, and sclera are all going to be affected. Albinism is going to be pleiotropic because it affects pigmentation. So you have optic fiber development affected as well because there is pigmentation associated with your eyes. So we have consanguinity. Consanguinity is a coefficient of relationship. So you have different degrees of relationship. So your degrees of relationship are first degree is your parent and offspring. So you're a first degree to your parent and you're a first degree to your sons and daughters. Second is going to be your siblings. I really messed that one up, sorry. So your siblings. Your third is your aunt, uncle, niece or nephew. And your fourth is first cousins. Okay, so you have consanguinity, which is going to be calculated by a coefficient of the relationship. So I have it here. So when you're calculating consanguinity, you are going to begin with one individual and proceed up the pedigree to the common ancestor, and then down the pedigree to the other individual. The coefficient of relationship for one route is given by 1/2 n minus 1, and n is the number of individuals in the route. And then if there are multiple routes, multiple common ancestors, then you add those coefficients together. Okay, so for this one, for example, you start here and you're going to go up. I'll do a dotted line. And so here's 1. And then you go up to the common ancestor, and so that's 2. And then you go down, and you have 3, and then you have 4. And so your consanguinity is 1/2 n minus 1. And so it's 1/2 4 minus 1. Oh, sorry, is that 4? I should actually, 1, 2, 3, 4, 5. Sorry, 1/2 5 minus 1. That makes sense. And so 1/2 to the 4th degree is what the coefficient of the relationship is. Now, if these people weren't related, you wouldn't even get a coefficient of relationship. It would just simply keep going on and on and on. And so you can imagine that the smaller this number is, the more closely related the individuals are going to be. So one thing to keep in mind with consanguinity is the rarer the recessive disease, the more likely the parents of an affected individual have some sort of relatedness, kinship. Cystic fibrosis, classic galactosemia are both usually connected to some form of consanguinity. Cystic fibrosis is 1 in 25 out of European white population is a carrier, and therefore you have a 1 in 25 chance of meeting another carrier from the general population. This drops to 1 in 8 if you are intermarrying first cousins. So remember, first cousins is that fourth degree of relatedness. Galactosemia frequency is 1 in 170 or 1 in 8 if they are first cousins. So the probability of bringing recessive traits together through consanguinity increases greatly rather than marrying out into the population. So consanguinity overall is going to increase frequency of genetic diseases. The closer the degree, the greater the increase. Okay, so next, part 2, we'll talk about sex-linked and other modes of inheritance.